Thanks all for showing up. Clearly the deceptive title has drawn you all into my trap. But there will be some sex and there will be some drugs, so don't worry about that. You'll get some of that. Might not be quite as sensational as you were hoping. As you just heard, I work at the Royal Botanic Gardens, Kew, so it would be remiss of me not to do a little bit of advertising about Kew Gardens. What a fabulous place. I'm sure many of you have been there, maybe not as recently as you perhaps should, so do come. You're very welcome. Many people know the gardens as being a wonderful place to spend time, a beautiful landscape, lovely gardens. We've got about 300 acres. It's got lots of old buildings. It's a World Heritage Site. What you may not realise is that we have over 500 plant and fungal scientists doing really important work addressing some of the global challenges. And I'd like to talk to you today about some of the work that I do, some of the interests I have. So we have about 500 scientists, but we also have we have over 100 species of bees too. And that's another passion of mine, working with and trying to support bee diversity. So here we have a little bit more about Kew, our mission to understand and protect plant and fungal diversity for well-being of people and the future of all life on Earth. And we do this around two main areas. What we see as the twin challenges of climate change or global change, because of all the other changes that are coming with it, and biodiversity conservation. We don't think you can really solve one without the other. So we address both of these issues together as part of our manifesto for change. And within that, we have different areas which allow us to deliver this mission. The first is delivering the high-quality science, some of which I'm going to share with you shortly. But we also try and inspire people. The visitors that come in, we don't just let them wander around. We grab them and tell them fantastic stories about the work that we're doing and why it's important, and try and engage them, especially as young as we can, to get them as enthusiastic about plant and fungal science as we possibly can. We're also training more established young scientists through our capacity building and teaching programs. We also do our best to extend our reach or speak to the media and speak to groups like yourselves to share our knowledge and inform and influence. And ultimately, we want to be able to influence policy development through engagement with politicians and other similarly influential people. So one of the most important parts of our infrastructure at Kew are our collections. These are the historic collections that we've built up over 200 or 300 years now, perhaps the most famous being the herbarium, where we have over 8 million specimens of plants. We now have a fungarium containing about 1.5 million, and these are really important resources for the research we do. We also have lots of living collections, so we have the living collections in the gardens. We have about 17,000 different species of plants, making Kew arguably the most diverse place for plants on the planet. We also have the Millennium Seed Bank down at Wakehurst, which actually is the most diverse place on the planet because we've got 40,000 species basically in a hangar. But we have lots of other interesting collections too, including our library, our books and economic botany and so on and so forth. And this really underpins the research that we do. It's a really important part of our infrastructure and it's open to everybody. We're now digitizing it, so you will be able to access all of the information we've got at Kew yourselves through our open portals as soon as we've got it all done. We're nearly there with the herbarium. So today I'm going to talk to you about my own interest, which is really around the chemistry of the interactions between pollinators and plants. And I always find it a little bit of a struggle talking about chemistry. We're all very passionate about biology. It's always the chicken and egg question. What came first, our passion for nature or these heroes that have shared it with us over decades? But we're all very happy to watch hours of TV about biology. And even these days, physics has become quite cool as comedians join scientists to get excited about the stars and gravity and other fantastic things. But when it comes to chemistry, we have a very different view of the world. Chemists are always maniacs. We've got Walter White here, the errant chemistry teacher from Nebraska who went off into the desert to synthesize methamphetamine, or when we've even got a great platform on the telly like Peter Wathers from Cambridge University doing the Royal Institution lectures in 2012. He made it more like Hogwarts than real chemistry. So today what I'd like to do is to take a little step forward in helping you appreciate chemistry, notwithstanding some of the skeletons in the cupboard that chemistry has. I'm not suggesting that it's a completely innocent topic, but I would argue that biology and physics are more dangerous. And actually chemistry is more likely the cure. And hopefully today you'll be believing that actually chemistry can be the cure in lots of natural circumstances too, particularly when it comes to pollinators. So as I said, I'm a chemist. I'm interested in chemicals. I have a laboratory at Kew Gardens where we study plant and fungal chemicals. We're also able to undertake very interesting biological assays with different organisms. My area of interest is bees, particularly bumblebees. They're a great representative of wild pollinators, but they're quite easy to handle in the laboratory, which is an important thing to do. We buy most of our bees from Copper or Biobest, who obviously provide them for tomato and strawberry growers. But we do also sometimes collect them from the wild too. We're able to study in our laboratories and in the gardens too. So today's talk is going to be about chemistry. I don't want you to be scared. Chemistry's fine. It's not rocket science. It's quite simple. All the chemistry that I'm interested in is really about chemicals that are comprised of just carbon, hydrogen, oxygen and nitrogen, and mostly carbon, hydrogen and oxygen, to be fair. And you'll be seeing lots of chemical structures. You don't have to know what they all mean, but just if you're interested, I thought I'd put this one up here. One of the things we do when we draw chemical structures is try and avoid the chaos, and we convert things like this to this, so we don't have to write lots of letters everywhere. So when you see a six-membered ring like that, that's essentially 6 carbons with hydrogen sticking off them. It gets messy otherwise, and if you've got a row of carbons like this, we'll just draw it as a line. So just to kind of give you an idea of what these chemicals mean, and just to make sure that I get you over this hurdle of being freaked out by chemicals, here's a whole bunch of them, but they're actually your friends, really. And you can see that they all differ enormously in the shapes, and essentially the reason why chemicals are different is because the different positions each of these atoms sits in that molecule. But that makes a huge difference to what they do and why they're important. And just to give you an idea of why these are important, if I give them names, they'll seem an awful lot more familiar. I expect all of us have had some of this today. This is just caffeine, okay? We've had a shake of the head there, but I'm sure many of us have. We've got aspirin, we've got morphine, we've got anti-cancer drugs. These are all the array of kind of chemicals that we get from plants. So they're all very familiar, but I will be showing the odd structure as we go along. One of the other reasons this slide for me is important is it shows the range and diversity of compounds and why they're so important to us, to people. And in fact, we have many, many drugs that come from plants, or at least have their origins in plants. We might then adapt or modify them with different chemical structures to make them more effective at the thing that we want them to do. But here are some examples. I'm sure you're familiar with them. Cinchona bark, the source of quinine, which was the first effective treatment for malaria, now used as a prophylactic still, or at least as the chemical basis of it, although in fact increasingly we're now using another chemical called artemisinin, which comes from a Chinese plant called Artemisia. So again, from a plant. Taxus brevifolia produces a compound called paclitaxel, which is a really important treatment for some cancers, including breast cancer. My great friend Sandy Knapp at the Natural History Museum wrote a fantastic story about how this saved her life. So these are compounds that just come straight from plants, and we use them in medical applications. Madagascan periwinkle, a source of vincristine and vinblastine, a really essential treatment for non-Hodgkin's lymphoma and some other leukemia type treatments. So a whole host of different chemicals we get from plants. But plants don't produce these chemicals for us, of course. They produce them for themselves. They don't really care what goes on with us. They want to survive. And this is really the reason why they are so chemically diverse, because they can't move. Plants are stationary, they're rooted in the ground. If they're attacked, if you're chased by a lion, you run. Trees don't run, they have to do something else. And of course, they develop all these various defense mechanisms. Most of us are familiar with being stung by a stinging nettle or a cactus spine, like this giraffe maybe down here eating an acacia. But actually, most of the defense chemicals that plants produce are chemicals like the ones I showed you. They're toxic to would-be herbivores, or they might be distasteful, or they have some developmental effect. But there are also challenges for plants that can't move when they want to get their pollen from one place to another, from their anther to a conspecific flower. And of course they do that by recruiting the services of pollinators, or at least 90% of angiosperms do. They also have a job dealing with herbivores by attracting mutualists like parasitic wasps, and they do this again by attracting them with smells that those wasps find familiar. And this whole process is all chemical, and this is my world. This is where I study chemicals and try and understand how plants are driving these interactions and taking control of the fact that they cannot move. So my focus is really on animal pollination. We're all very familiar with that in this room, of course. We know it's about pollinating different flowers, taking the pollen from one to another. Your best friends, the honeybees, are fantastic at doing this job. And it's a really important part of our food production systems. There are estimates that its global value is at around $600 billion from work by Simon Potts at Reading and all. And they also are very important for producing the healthy stuff, the fruits and the veg that we eat. So they're a really important part of our diet. But also, because they're important in allowing our non-food plants to reproduce, they're a really important part of supporting biodiversity, and they're really essential for conservation, which is why they're a really important part of the story for Kew. So here's your friend, Apis mellifera, my friend too, don't worry, I like honeybees too, although I'll have something to say about them at the end that might be a bit controversial. So, but of course, honeybees are not alone. They share that world with 20,000 at least other species of bee, all fabulous, different shapes and sizes and colours. And as I inferred at the beginning, at Kew we're particularly interested in this diversity and have so far managed to identify over 100 species. That's more species in Kew Gardens, by the way, than they have in Ireland. So actually it's a really interesting example of what you can do in a city, and one of the things I'm quite keen to do is do more about supporting biodiversity in cities. And again, I'll talk a little bit about that towards the end. It's something that we can do to engage more people in understanding what biodiversity is. And with bees, actually, they're a really good tool for doing that, because I think you'll all appreciate this, but most people have come on board with the idea about bees being a good friend to human beings. It's not that long ago where they were still making terrible movies in Hollywood about killer bees, 20 years ago. But actually we've come an awful long way, and I think they're a really good entry point for people to understand the importance of biodiversity and why it's important to conserve it. But we have a problem. We have increasing evidence that pollinators are in decline. Not all of them, actually. Bombus terrestris, just as an example, is actually doing pretty well, probably because it's a generalist. Perhaps some of the specialist bees are having a bit of trouble, but also other pollinators like hoverflies are also struggling and having challenges. And there are multiple factors that are influencing this. In this rather convoluted figure I've got, you can see I've separated them into the anthropogenic drivers and the natural processes that influence decline. This is partly because I've avoided all the anthropogenic drivers, because there's plenty of people working in that space. There's not so many people working in these natural processes, and so I figured that was kind of a more interesting and a rich seam of research to undertake my own work. But there are a multitude of things pressing on pollinators, causing these potential declines in different parts of the UK and elsewhere in the world. The ones I'm particularly interested in are these nectar and pollen chemicals here, and they actually can influence directly pollinators, or they can influence other parts of the natural processes that are important for pollinators, and not necessarily a negative way. And what I'll try and show you today is some of the benefits that we might get from understanding more about the nectar and pollen chemistry. Or in fact, today I'll just be talking about the nectar chemistry, actually, because I don't have enough time. If you want to read more about this, then there's an academic tome that we produced with the Philosophical Transactions of the Royal Society, where we talked about the natural processes influencing pollinator health from chemistry to landscapes, where we have lots of really good examples of how natural processes can be used to better understand how we can perhaps reduce the impacts of some of these processes. Okay, so today I'm going to be focusing on nectar chemistry. You'll all know nectar's so important because it produces one of your products, the honey. It's a lot of sugar, different types of sugar. You'll probably be familiar with different proportions of the glucose or fructose or sucrose makes different honeys. The bit I'm interested in are the other chemicals that you get in there. And it may be a surprise for some of you to learn that actually most nectars contain quite a range of different chemicals, and they're perhaps partly responsible for the different flavours that you get with some of these honeys. They're certainly responsible for some of the interesting effects that we've been working on at Kew. And there are three examples I've got here. Mediating pollinator specialisation, number one, and that's to do with that compound granotoxin, which I'll come to. Antimicrobial activity, nature's pharmacy, and that's calanine, which is in the middle, and then I'll be talking a little bit about caffeine at the end because of its ability to modify pollinators' behaviour. So, first off, nectar toxins mediate pollinator specialisation. And this is work that actually stemmed from, well, I'll come to that in a minute actually, but just to kind of introduce this idea of specialisation if you're not familiar with it. Some plants develop relationships with animals that become very specific. And the reason they do this is because you can be assured of pollination service provided your pollinator is around. Perhaps the most famous example is this famous orchid, Angraecum sesquipedale, that was brought back to Darwin. And he looked at it and thought, that really long nectar spur, I bet there's a really massive moth somewhere in Madagascar that feeds off this. No one had found it, and 40 years later, this moth species, Morgan's sphinx moth, was found. And of course, it fits perfectly, the length of the proboscis. And this is a specialist relationship, so nothing else can get to this nectar, only this moth. So when this moth is around, it only feeds off this orchid. So every time it feeds, it's going to the same species, a conspecific. So it's always taking pollen to another flower of the same species. So that's really ensuring that that plant gets pollinated. Of course, the converse is if that insect isn't around, then that plant doesn't get pollinated. So it's a little bit of a balance. But this is what specialization is. And you can see it throughout the pollination world with these different animals. So there's a lovely picture that I managed to capture. I don't know how I managed this, but this is another sphinx moth in South Africa feeding off Plectranthus. And you can see how the architecture of the flower fits perfectly. The long proboscis and the anthers tickling the hairy chin so that when it visits another flower, it'll transfer that pollen. And even with hummingbirds and sunbirds, you'll see these same things where the anthers come out and just cover the belly in pollen. There are already some chemicals that you'll probably be familiar with that actually drive some of these pollinator interactions. The colour blue, for example, which comes from delphinidin. Delphinidin blue is actually an intrinsically attractive colour to pollinators. And then some flowers are very clever and they produce chemicals that deceive pollinators. So this example here, we've got this hammer orchid, Drachea, which looks a little bit like, not much, but a little bit like a female thinid wasp and produces the sex pheromone of the thinid female wasp. So males come and think they're going to mate, and then they get a pollinia stuck to their head, fly off and visit another one, and then transfer that pollinia. Amazing, amazing nature, really quite, and in fact, in this particular case, it's so effective, things can really get out of hand. We were interested in this because we were studying a really big problem in parts of the British Isles. I'm sure many of you are familiar with rhododendrons being a problem, weed in many parts. Nothing, I mean, if you've seen it bad and you haven't been to Kerry, you need to go to Kerry to see how bad it is. Kerry in the west of Ireland, it's a perfect landscape for rhododendrons. And so we were interested to know, what does this plant do to bees? We knew it produced some weird chemicals because there's a very famous honey called mad honey. It's called mad for a reason. If you eat it, it makes you go a bit loopy. So we knew that there was something in the nectar, and it turns out to be a group of compounds, particularly this one called granotoxin. But we didn't know what they did to bees, and we thought it would be really important to find out what they did to bees, because if they were also nasty for bees, then that's even more reason to get rid of them. And it's obviously a huge story in Ireland. You know, they've had debates in Parliament and so on. The story of this mad honey, though, is worth just dipping out of my own work for a moment, if you haven't heard of this. But we've known about mad honey for a very long time. In fact, this particular guy, who is arguably the godfather of plant chemistry, I should look up to him and semi-worship. He was a guy that studied plant chemicals without the equipment that we have today, largely by making his subjects eat stuff to see what it did. To be fair to him, he also ate quite a bit himself and actually ended up killing himself with them. But so he was very interested in poisons in plants, and he knew all about mad honey. And actually around this part of Turkey, you get quite a lot of this mad honey. This is where Rhododendron ponticum comes from. And when he heard that Pompey the Great was invading with his armies in 66 BC, he put all these honeycombs down along the path, and as the soldiers walked in to Asia Minor, they thought this was gifts from the gods, so they ate the stuff up, all fell into a drunken stupor in their tents, and while they were in that drunken stupor, Mithridates walked in with his army and slaughtered them all. So perhaps the first example of a chemical weapon of war, all down to some nasty honey. Lovely little story. But so we did some work to see, right, let's find out what these chemicals do to bees. And I have to say, they're not very nice to honeybees. British honeybees, at least, you'll have noticed that honey makers, the honeybees in Turkey are actually adapted to this, but not in the British Isles. So we showed that actually the compounds were quite toxic to honeybees. And in fact, if they're given other food, they will avoid it. They will avoid these toxins. But we were really surprised to find out that Bombus terrestris was absolutely fine with it. And in fact, if you've ever noticed when rhododendrons are in flower, you'll almost exclusively see bumblebees feeding on them. And you'll very rarely see honeybees going anywhere near them, at least not in this country. And it made us think, well, that's interesting, because normally if you've got an insect toxin, it's toxic to insects. And here we've got this kind of, this difference in their susceptibility, it's selectively toxic. And we thought, well, what could possibly be going on here? We also found that actually where we had lots of rhododendrons, especially at that time of year you get a lot of Bombus pascorum, you actually get lots more Bombus pascorum nests where you've got rhododendrons. So actually rhododendrons are good for bumblebees. Probably not the most popular thing to be suggesting to an ecologically minded audience, but it does look like they're actually quite good for bumblebees. So maybe we need to sort of think about it when we're wanting to clear vast areas of land. I saw a big clearance in Richmond very recently where they took out all of the rhododendrons, and you just sort of wonder what happened to all the bumblebees then, because they didn't have their food source. So we then had a bit of a think about this and thought, well, if it's selectively toxic, could it be having some kind of filter system? Is it that the rhododendron don't want honeybees to come and feed off them and just want bumblebees? And it turns out that this might be the case. And we've done work on the whole plant group of rhododendrons, where we've assessed their what we call pollinator syndrome. This is the way we measure what kind of pollinators those flowers like. So as you saw with Darwin's orchid, when you've got a long nectar spur or a long corolla, typically that would be adapted for a long-tongued moth. So for example, this rhododendron species here is adapted for long-tongued moths. Here we've got yellow flowers, normally closed, requires a beak to open them and to access the nectar, adapted to bird pollinators. So this is where rhododendron's using morphological adaptations to filter out the pollinators it doesn't want. And as you get down to these kind of generalists, the open flowers, we find that these are always the species that have got the toxin in them. So we think it's actually a mechanism by which these plants are filtering out their less preferred pollinators. And that we think is the case in the UK. So that's an example of where these compounds can drive specialization. But where I think they're probably most useful is where we think they might be able to help reduce disease burden in bees. And that's the next topic I'm going to talk to you about. This is work that I've been developing over the last probably near decade with some very good colleagues of mine, Hauke Koch, who's now, he's a German scientist, but now based in Australia, and Mark Brown, who I'm also still working with now on a project I'll talk about shortly. And we published a paper in about 2017 where we considered the importance of plant chemicals in driving or influencing the transmission of disease in bees. What often happens is bees transmit and contract parasites when they visit flowers. They defecate in flowers, and then they come and feed at flowers, and they pick up those and fly off. And in fact, you get even across species. So there's evidence that honeybees transmit to bumblebees, and it probably goes the other way as well. And of course, those diseases affect foraging behaviour. They affect queens found in colonies, in bumblebees, a whole host of different problems, and especially when you have additional contributing factors like pesticides, they exacerbate those problems. But what we considered was that the fact that we have these chemicals in nectar and pollen, that maybe they could influence those diseases. And they probably do, because they need to protect things like nectar from yeasts and bacteria that might otherwise waste that sugar. It's a very expensive resource. So we've done work basically in this space. We thought we'd look at plant species that were helpful to us, and those plant species are the ones that we get monofloral honey from, because collecting nectar is a real pain. I don't know whether you have tried it yourself, but each Calluna vulgaris flower has between 0.1 and 0.5 of a microliter. And so the fact that honeybees are very willing to help us with our experimental work by collecting kilos of the stuff is a great help. But also, Calluna heather is a very important landscape species, providing I think it's the second or the third most important nectar producing plant in the UK, but it's in decline in the south of England, and I'll come to that briefly. So to start this work, we used honeys. We tested a whole bunch of different monofloral honeys. The one I'm showing you here is the one that was most effective, and this is Ling heather. This is Calluna vulgaris heather honey. And this is essentially a measure of how much this honey prevented proliferation of Chrysidia. That's this organism you can see in an electron micrograph, Chrysidia bombi. It's a really important parasite of bumblebees. It lives in the hindgut, and at high levels it effectively neuters queens, stops them producing colonies. And it can cause morbidity as well. We don't know all of the problems it has, but it's also quite a useful model species. And we tested a whole bunch of different monofloral honeys. Erica, we've got Malus, we've got Tilia species, I'll come to them later, Echium vulgaria, a whole host of different monofloral honeys. But because Calluna heather was the most effective, we thought we'd look a little bit further at this. So we did some work. I found a chemical in it, cut a long story short, because that is a very long story, finding these chemicals, called Callunine. We've called it after Calluna, which I think is very sensible. It's this compound. It's quite simple, but it is very biologically active against this parasite. And it is biologically active at concentrations at which we find it in the crop of the bumblebee and at concentrations at which we find it in the nectar. So this is a chemical that's in the nectar that's biologically active against a gut parasite of bumblebees, and it's a plant that those bees feed on. So it's a natural pharmacy for these bees in this landscape, or we think it might be. We don't yet know. We know for sure it's biologically active. We know a little bit about the mechanism as well. If you can see this wriggling, it's quite hard. If you've got a small screen, just here, you might be able to see this little thing wriggling. This is the parasite, and this is the gut wall of the bee. And you can see that it's locked in there with that, what we call flagellum, that little tail that it had. So it puts that tail into the gut wall and then sits there in the gut, taking all the nutrients as they come through in the bee and then reproducing. Over here, you can see we found what the effect of this compound is on the parasite is to nip off this flagellum. So where we have them treated, they don't have a flagellum. They just literally pass through the bee without being able to anchor. So they can't attach, whereas in the untreated ones, they all still have a flagellum. So we think that's the mechanism. Okay, so we've got this chemical that's occurring naturally in the landscape that bees are feeding on naturally that helps them reduce the disease burden of the diseases that are most important to them. So we've got a new project, as I said, with Mark Brown, where we've got support from the UK Research and Innovation, where we're looking to understand whether this actually happens in any measurable way at landscape scale in the field. It's important work because, as I said to you already, we're losing lots of Calluna heather. It might be that we're replacing it with oilseed rape, but my view would be if oilseed rape isn't producing the same kind of quality nectar that Calluna heather is producing, then we're not replacing like for like, and it's important to be able to know that. So we're losing a lot of our lowland heaths in the UK, so it's an important target. And in this project, we're looking to determine the effects of callunine and other metabolites actually in the nectar, because we've got lots more work to do in those honeys. And then we're going to try and see if we can demonstrate whether it reduces epidemics in colonies across the landscape. If it does, then we're able to advise and inform DEFRA and other landscape managers about the importance of Calluna heather. So just a quick look through some very recent work, but this is our postdoc, Lachlan Jones, who's been doing some work recently, where we've been trying to test to see what these compounds do in colonies. We've only been working on individual bees at the moment. So what he's done is collected queens from the field, so these are wild queens, rears those colonies to about 10 to 15 workers, and then he infects 10 bees from each of 3 colonies with 25,000 cells of that Chrysidia. So they've got an established infection in that colony. And then he starts treatment a week later. And then after 10 weeks of treatments with callunine and also an extract of the honey, so it has all of the compounds in there, we then monitor the the feces of the bees after about 10 weeks to see, in fact, throughout the whole of that testing period, to see what the effect of these compounds are. And it turns out that the proportion, this is a little bit misleading because this is the proportion of bees in those colonies with Crithidia, which you can see is less in the heather nectar fraction and the calanine, but not as much as we would have liked. But actually, when we look at the proportion of bees that are infected or have flagellated Crithidia, i.e. the Crithidia that have got a tail and so can infect, it's less than half. So where we have colonies that are feeding on nectar or artificial nectar that contains this Calluna heather compound, they are benefiting actually to a really significantly measurable effect. And this is likely to have a much more important benefit or effect on the ability of that colony to survive and produce new queens for new populations going forward. So we have the first signs that this might be a real benefit in the landscape. We also have evidence that actually we're getting better queen survivorship for queens feeding off these compounds. So there look to be real good benefits of this stuff at colony level 2. The next step is to try and see if we can find out whether diseased bees in our landscapes are healthier, or if we can put bee nests in our Calluna landscapes, whether they have a lower uptake or acquisition of these parasites. That's not the only story. You'll recall that we had lots of other honeys, and I've got 2 more examples here. I'll only touch on them briefly because I appreciate we're a bit short of time here. But I've got 2 honeys I'd like to talk about. One is Arbutus, which is the strawberry tree. Anyone familiar with the strawberry tree? Yep, lovely. And Tilia tomentosa, which is the silver lime. So these both produce monofloral honeys that are also biologically active. They reduce the growth of this parasite. My brother lives in Sardinia, actually, where they have this, they call it corbezzolo, and they use it like it's a medicinal honey, actually. But I think mostly because it tastes quite horrible. If it tastes horrible, it must be medicinal. I think that's the logic. But they treasure this. But it's kind of nice that now we've been able to give them a little bit more substance and evidence for their claims. But I'll first talk to you about this one. This is Tilia. Tilia, of course, the lime tree. A huge amount of nectar production around June, July in the UK landscapes. A really, really important tree for pollinators, for honeybees, but also for bumblebees and lots of other species of pollinator that require those food sources. And we found a similar effect. You can see here we've got this effect. As you increase the concentration, as we were showing with the other, you get much reduced cell growth of this parasite down to concentrations at which we find these compounds in nature. But the strange thing about Tilia is actually the compound we find in the nectar is this one, which has got this big lump of sugar on it. Plants often stick sugars to toxic compounds to make them less toxic because they become more water soluble, so they can travel through cell membranes and pose less of a risk, and they can be compartmentalized in the vacuoles in the cell, so they're kind of a little bit easier to handle. And then obviously they get released into the nectar where, obviously having a bit of sugar on it helps it to be soluble in the water, the watery or the aqueous environment of the nectar. So this is what occurs in the nectar. So it's not actually active in the nectar, but as it passes through the insect's gut, the insect has these sugar enzymes that cleave off this bit of sugar, leaving this terpene. And this is biologically active and what causes this. So this is another little kind of side story to this. Things might not be as they seem. You may not have the potent activity in the nectar itself, but you need to study the organism in a little bit more detail. So the next example, Arbutus, is even more weird. So it occurs in the nectar as a biologically active compound. This is called unidone, after the plant, Arbutus unedo. And it is biologically active, and you can see again here, this is the active one here, causing a reduction in growth of this parasite. But we think it might be a bit toxic to the bee. So as it passes through the gut, the bee glycosylates it, sticks this sugar on it. Then it ends up in the hindgut where the parasite lives, and then only bees that have got a healthy microbiome, That's the good bacteria that we all know is important for ourselves, but it turns out are also very important for bees, where that microbiome then reactivates it to be biologically active against the gut parasite. So this is an example of where a healthy gut microbiome in those bees is an important part of enabling those animals to benefit from those landscape pharmaceuticals, if you like. So a really nice little story there. And one other thing I should add, we are, and I'll come to this briefly at the end, we are doing a lot of work now on trees that are better suited to our landscapes, not just today, but 50 years' time. And actually, I think both Arbutus, unedo and Tilia are going to be the trees that we're going to have in our landscapes in 2100, when all of our beeches and all of our other trees have all died away because the temperature's 4 degrees centigrade higher and the weather's gone weird. So anyway, so there we are. There's examples of our natural medicines for bees. So the third story I promised you was on the nectar toxins that manipulate pollinator behaviour, and this is where the drugs comes in. Well, at least we like to imagine that it's plants drugging bees to do stuff for them. One thing you'll notice about this picture is this bumblebee. This is Bombus hortorum. For those of you who don't know it, it's a long-tongued bee, as you can see. It's got a very long tongue, but it's a nice image because what it shows you is that bee is approaching that flower knowing what's coming. It's remembered the signs. It's either seen spots on the flower or it's smelled the odour, something about that experience before it's landed, before it knows there's food there. It knows there's food there because it's got its knives and forks out. It's ready to eat. It's got its proboscis out. It's saving time. It's landing with its proboscis out, straight in, get the nectar, get out. Very efficient, obviously, foragers, as you'll know from honeybees. So a really good example of how bees use memory as part of that experience they use to be more effective and efficient foragers. And that's an important part of this story because we found nectar in some plants, including coffee and citrus, contained caffeine. And it might not come as a surprise to you to find caffeine in the nectar of coffee, apart from the fact that the reason plants produce coffee is because it's toxic. It's a toxic compound to insects particularly. That's why you get so much of it in coffee beans, up to 2 or 3, 4% by weight. Loads of caffeine in there to stop beetles and other insects eating that fruit. And so to find it in the nectar was a little bit of a surprise because that's the gift for the pollinator. That's what the plant is giving to the pollinator to visit. Well, it turns out that it's at too low a concentration for the bees to taste or detect, strictly speaking. Bees can't detect it, but it's still there. And we were fascinated to know whether this had any other effects. And we have this interesting little assay called a proboscis extension response. So this is us feeding a tethered bee. No bees were hurt in the making of this movie. You can see a tethered bee, and what we're doing is we're feeding it with a liquid nectar, touch the antenna, and at the same time is we're blowing an odour across those antennae, just for about 6 seconds. So that bee is feeding, and at the same time is sensing an odour stimulation. If you do that about 6 times, only 6 times, the bees will remember that. And then in this next video, which is the green one, you'll see a bee. And when the red square comes, because of course I can't show you chemicals, when the red square comes, that's when the odour comes. And you'll notice there's no food around at all. So here's a bee tethered, and then in comes the odour, and out comes the tongue. So we've trained that bee to stick its tongue out when it thinks it's going to be fed. And this is a really useful tool for measuring, say, the negative effects of pesticides, which do affect memory formation. So you can actually quantify the effects of these negative things. But in this particular case, as is shown in this figure down here, we found that caffeine actually improved bee memory over time. So the proboscis extension response after 24 hours, you can see, was much stronger with bees that were feeding off caffeine at natural concentrations. And there's been much work since that has shown this is actually a behavioural effect. There's a team at Sussex who did work with honeybees at colony level and showed that they were more effective at their waggle dance. And they were, it's an amazing thing to think that this compound, caffeine, is in the nectar and is effectively making bees remember odours associated with that flower. And it does make sense, because if a bee is going out foraging the following day, and the first thing it does, it smells something it remembers as food, it's going to go back there and pollinate it. So it's a really clever and crafty tool that these plants have, and it seems that this sort of popping up quite a few times in nature. And actually, we did a little bit of neurophysiology and showed that actually there's a pharmacological effect in the part of the bee's brain where they form these associated stimuli memories. So there is a pharmacological basis for this. And now what we're trying to do is actually drug bees. So one of the problems strawberry and tomato farmers have is getting bees out of the nest boxes, and when they come out of the nest boxes, actually visiting the flowers of the fruit that they want, or the flowers they want pollinated, rather than just trying to escape all the time. And so we've been working with farmers in Kent to try and develop a little device, this kind of funny thing here, which essentially contains caffeinated nectar and a little odour source that gives them the artificial odour of the strawberry flowers, or we're starting to work with tomatoes now, to try and get them to be more effective pollinators. So it's a way of kind of using curiosity science to influence how we then are able to improve the pollination services of the bees. It might also explain one of these long-standing quandaries or queries that we've had in nature. For hundreds of years, we've known that tilia trees are often found with dead bees underneath them around June, July. And no one's ever really known why. There's been some work suggesting that it's because of toxic sugars that the bees can't use. Actually, this isn't the case. The guy that did that, a Brit in the 1970s, I don't think had very good chemical equipment, and we've disproved that now anyway. But what we have found, and caffeine does pop up in very different unrelated plant species or plant groups, what we found is that caffeine occurs in the pollen of tilia. And it might be that the bees, they're eating a little bit of pollen, they're getting caffeinated, and they're associating that with tilia, and they're beginning to overvalue it, and they're returning to the tree even after it's stopped producing nectar, but still smells. And so they're coming back and they're flying around the street thinking, I'm sure this was where I got food last time, until eventually they just fall down exhausted. And that happens to bumblebees quite easily, actually. So it may be that we're able to explain this really strange phenomenon with linden trees. I'll come back to that story in a bit, but you can see this bee struggling on the ground after having fallen down, being a bit exhausted. So just in summary, before I wanted to capture a little bit of another story while I've got you, just in summary, we've got nectar metabolites that mediate plant-pollinator interactions. We've got nectar metabolites that could inform landscape enhancement for pollinator health. And then we've got nectar metabolites that could be used to optimize commercial pollination, all based on compounds that these plants are producing naturally in their nectar, probably to enhance their pollination, either by protecting it from microbes or manipulating the pollinators that visit. One thing I wanted to talk to you about now as well that relates to this is messaging. Now in the paper, in fact I'm just going to go back and show you this paper, we had a title here which was Do Linden Trees Kill Bees? Reviewing the causes of bee deaths on silver linden. I really regret that now because the answer to the question is no. Linden trees don't kill bees. Bees die because they're a bit too enthusiastic for the nectar, but they're not being poisoned by it. Nevertheless, when the messaging is wrong, you get problems. And this is a little bit of text from a transcript of the House of Lords Hansard from the 19th of June 2018. And this is Baroness Bloomfield of Hinton Waldrist. She's talking about bee-friendly plants. I suggest that we avoid Rhododendron ponticum, whose nectar is toxic to bees. Well, honeybees, fair enough, but they don't go to visit it anyway, and it's good for bumblebees. And the silver lime, Tilia tomentosa, which uses caffeine to trick bees into visiting empty flowers, whereupon they die of starvation. So clearly the message didn't quite get through, and the important thing to remember about Tilia is it's a fantastic resource for all sorts of different pollinators. But the messaging needs to be really clear, otherwise it gets misinterpreted. And this could have been disastrous. And at the bottom here, we've got Lord Stevenson, no relation of mine, by the way. I'll have to go around my garden eradicating Rhododendron ponticum, which I've just planted in large numbers. Must be a very large garden. But anyway, I think it's really important. I think it's a really important thing. And of course, one of these relates to honeybee keeping, of course. You know, I think there was a confluence of different stories in the early noughties. We had this colony collapse disorder problem in the US that was a massive issue. At the same time, we started getting evidence of pollinator declines in Europe, in Northern Europe. And the press just found it all too complicated, so shoved them together and said, save bees, which then distilled down to save the bee. And as a consequence, we've got lots of people who are really minded to be kind of environmentally sensitive and do the right thing and do things that help biodiversity that are maybe not doing what's required. And in about 2018, Richard Glassborough, some of you will know, came to us and said, look, I think we've got a problem. Can you help us apply some science to this issue? There's obviously a rising awareness of environmental harm. There's a fascination with bees, but with the oversimplified messaging, he was aware that there was an excessive increase in honeybee keeping in cities. And they had solutions, of course, but they wanted to have a stronger message or evidence to support this. If you look at London from above, there's nearly 50% green space, but actually not all of that is really suitable for forage. Perhaps only half, because lots of it's playing fields, which is great, of course. We're very lucky to have such a wonderfully green city, but not all of it is really suitable for pollination services. But we did do this work for Richard, and it was kind of really trying to address this issue about the intensity of beekeeping in cities. Clearly, you can see here, we've got 12 hives or something like that on a rooftop. And by our calculations, London's landscape can support about 7.5 hives per square kilometre. And these hives have not got a square kilometre of green landscape. They've got a small rooftop. So it really is potentially a bit of a problem. We produced quite a lot of strong evidence here, where we overlaid the green space based on some generous estimates of what that could support in terms of pollinators, or at least in terms of honeybees. And all the pink and light pink areas are where honeybee keeping is unsustainable in London. This is 2020, and it hasn't stopped yet. And we still come across people who want to do their bit for the environment by putting a honeybee hive on their loft, on their roof, and it's not really helping. And in fact, we've often kind of argued that there's this kind of potential competition for food resources between honeybees and wild bees. And there's been some recent work that has demonstrated this. Is some very crafty honeybees robbing a megachilid of its hard-worked and hard-earned pollen. So we do have an issue here, and obviously people who keep honeybees are lovers of nature to a person, and people that want to keep honeybees are also lovers of nature, but we need to make sure the messaging is right and that people do it responsibly and that we don't overdo it in cities. Actually, you can see all around these sort of the edges of the cities, mostly it's absolutely fine, the numbers we've got. And I love my local produce from Wimbledon. We have lovely honey where I live. So, I'm not having a go at honeybee keepers, it's just we need to be sure. And in landscapes like London, where we have a wonderful biodiversity story to tell, we want to be able to tell that story as effectively as we can. We've got 140 species of bees that have been recorded in London. So really quite amazing. And what can we do? Well, we can leave our gardens a little bit untidy. There are limits, of course. This is probably a little bit too untidy. But rather shamefully, the people that own this garden, this is basically ivy, were fined £3,000 to tidy up the garden. And I would argue that actually that's a huge amount of important nectar and pollen resources around October, when honeybees particularly, and also this lady here who's foraging for nectar. This is the ivy bee, Caletes hederi, looking for food too. So I think we need to do a little bit more about making our gardens untidy. And we've actually had the good fortune to be sponsored recently by a London developer called Mount Anvil. I expect they're ticking a green box a bit, but if they're going to help support our research and we're able to do things for them that actually get good messages out, then we'll work with them, which is fine. And we've developed this blueprint for pollinator biodiversity in London, at least on their properties. They do these big builds, like 1,000 flats on land that's otherwise not being used effectively. But what they came to us after they'd heard the story about honeybees, and they said, well, look, we were thinking of doing honeybees. What can we do instead? So we're developing this blueprint where we try and get them to develop the green space around those properties with pollinator food resources, so we can support diversity and abundance of pollinators, making sure they've got a good habitat score. And by that we mean the untidiness, the places where those pollinators can have refuge or where they can nest and be undisturbed. We also have a site management score, which is basically no-mow, or no-mow may at least, and don't use pesticides or herbicides. But most importantly, and this is particularly important for Kew with our outreach programs, is engage with the public. So make sure that those landscapes around those properties are exciting and interesting for the people that live there. And so we're able to share that knowledge. And London is a fantastic place, like all cities. It's where most of us live. So if you want to have opportunities to get people on board with biodiversity conservation, that's where to do it. And so we're now doing a lot more work like this in cities where we're engaging people, but we need to support the biodiversity in those cities.
Should I add a shot of espresso to my beef fondant?
No. As long as it's pretty weak. The concentration in the nectar is less than you'd get in a cup of coffee. But, I mean, it wouldn't do it any harm. And if you put too much in, the bees wouldn't eat it anyway, because it would just be too bitter.
You were talking about different plants and the quality of the honey and the chemicals in it. Have you tried ivy honey?
I've not, actually. If you've got ivy honey, I would love to have some, because
You're very welcome
it's
to it.
chemically very weird.
It is. It's been tested in Trinity College, Dublin, and
Right.
they're telling me there's more properties in it than Manuka.
Right. Well, yeah, I mean, I think
It tastes like cough mixture.
Yeah,
so.
that sounds perfect for my lab. So I'll tell you what, so one of the things that I haven't told you about today, which we're doing a lot of work on at the moment, is looking at the nutritional quality of different pollens. And while for most of us we think of carbohydrate and protein as the key nutrients, the fats are really important too, sterols and fatty acids, and they get those, bees get those only from pollen. And we're doing a lot of work on sterols particularly. These are the compounds like cholesterol that we need for cell membranes and biochemical processes. Bees need them too. They get them from pollen. But they're really diverse across different plant taxa. And so each bee has a kind of a different sterolome, lots of different weird phytosterols. But ivy is the weirdest of them all. So I would be really interested if there was any monofloral ivy honey. I would love to have a look. Thanks.
I was wondering if you had any information on scabious pollen, because obviously there's a few solitary bee species that are obligate scabious collectors. And I think I heard somewhere that it has toxic qualities for other insects. I wondered if you had any information related to that.
Okay, well, certainly one of the things we find with a lot of the Asteraceae is that they have unusual sterols as well. And you're right, scabious, or Nautia arvensis, has a pollinator. It's our largest mining bee, in fact, isn't it? Andrena hartorfiana only feeds off. We think there might be a link between the sterols that the bees need and the sterols they're getting from the pollen. We think there may be these adaptations, dietary adaptations to the sterols that are in them, and that might be part of the explanation. If you look at the sterols of a bee, especially a solitary bee, a specialist bee, and then you look at the sterols of the pollen, they're almost exactly the same. We're not sure yet whether it's because the bees need those or they've adapted to them. But when you get an example like Andrena hartorfiana, which only feeds off Nautia arvensis, then it may be an explanation as to why they're doing that. And in terms of toxicity, again, it may be that other insects aren't able to use those sterols because they're all they are all unusual sterols in these plants. One of the things I think is a really nice example with that particular story, though it's a bit of a distraction from your original question, is it's a really good example of how we need to be mindful of how we understand landscapes, because there is also a nomada species that is a parasite of that Andrena species. And so if you don't have that scabious, you don't have the Andrena, and then you don't have the nomada species too. So it's a really nice example of that interlinking between floral diversity and the diversity of all the other organisms in that landscape.
Have you any suggestions for resources for people to understand the relationship between plants and pollinators? Have you got anything on Kew website?
Yes, if you go to the Kew website, you'll find lots of blogs about the work that we do, and we try and do that in a way that isn't talking down, but is sort of more plain speak. So there are lots of resources about this on the Kew website under the science banner.
If you could give us, say, 10 plants that would be ideal for bee health, not necessarily honeybees, but bumbles as well.
Yeah.
I mean, I'm thinking in particular of alliums. I try and grow a lot of alliums in the garden for them, and they seem to adore them. So it must be doing them some good. But
Yeah.
could you give us, say, 10 that would give us across the range?
Yeah, yeah. So in terms of bee health, I mean, I'm literally the cutting edge. No one else is doing this work anywhere in the world, apart from UMass, where they're doing some work with us. And we only have 3 species that we've published so far, and that's the 3 I've just talked to you about. I happen to know that bluebells are also bioactive against parasites, and even daffodils have some properties. We just haven't published that work yet, but we're coming round to it. But in terms of affecting the bee health at the moment, we don't have much more. I'd be very happy if anyone had any unusual monofloral honeys that they thought might help us do some work in this space. So a bit like the offer of the ivy honey, if anyone had any other honeys that they thought they were pretty sure were monofloral or semi-monofloral, and of course we can determine that by analysing the nectar and then comparing it to the honey to show whether or not it is from that source. But at the moment we don't know enough to be able to say, right, here's the 10 best plants. All I would say is that we do know that Arbutus unedo is great, Tilia is great, and Calluna vulgaris is great. Actually, I can also say that Erica's great. We've also done some work where we found similar compounds in Ericas, although that appears to be associated with the pollination or the pollinator. So we only have about 3 or 4 species in the UK. In South Africa, they have about 500 or 600 species, and they're all pollinated by different things like bird pollinated and moth pollinated. It looks like it's only the bee pollinated ones that have these medicinal compounds, which is interesting.
So have the cultivated species got the same benefits?
Yeah.
They have?
They should, yeah.
Do you think that a lot of the overpopulation of honeybee colonies in London, for instance, is due to misinformed companies wanting to tick that green box?
Yes.
Okay, that's well done. And the other one was going back to the bumblebees feeding on heather. Do you think they are truly self-medicating because they need to, or do you think it's just a happy coincidence?
Yeah, so we don't know this. It's a question I'm often asked, and we did try an experiment once where we took 7 bumblebee colonies that were clean. We took 7 that had this microsporidium called Nosema bombi. And then we had 7 that were infected with Crithidia bombi. So 2 with different diseases and 1 clean. And we let them forage wild down at Wakehurst Place, which is our botanic garden in the countryside. And we couldn't determine very much difference between what they were foraging on. But that might have been because there wasn't much in flower at the time. So I can't say conclusively, but I've just got a new PhD student that started with me at Queen Mary, University of London, who works with someone you may have all heard of. I'm co-supervising with a chap called Lars Chitka. You've probably all heard of Lars Bonkers Chitka. So that's going to be fun. But we're hopefully going to be able to answer this question. So come back to me in 3 years and I'll be able to say whether or not it's the case. But wouldn't it be amazing? Wouldn't it be amazing if they did? There is actually quite a lot of evidence about sort of post-ingestive behaviour changes where your body is doing something, not as necessarily as humans, but we can do these experiments with insects as well, where they're feeding on something else, not sort of consciously, but sort of their body is telling them, you need that now, you've had too much of that. So it's quite amazing. Yeah.
We as beekeepers are told that the more diverse the forage the healthier the
Absolutely.
The but then you get those species specific like Collegius hedera that only feed on one thing. So
it's really strange. I know it's an odd thing and quite how it works. Although there's a guy called Carlos Herrera who is a long-established pollinator scientist in Spain who recently contacted me and said I've found Collegius hedera feeding on these 4 different species of plants that are not ivy. It may be different, they're adapted in different locations. I don't think it's quite as clear cut. We've got a lot of good evidence over years where we've got where we can say that such and such is sort of monolectic, what we call monolectic, feeds only on one species. But actually, if push comes to shove, they might be a bit more flexible.
Well, also ivy is towards the end of the year when there isn't so much.
That's true.
Food.
Thinking about the antimicrobial properties, you'll be aware of the honeybee gut parasite Nosema. There used to be a treatment for that some moons ago, but it's not available anymore. I've heard that putting thymol into syrup can help with that, and as thymol is a thyme derived product. I'm wondering whether you're familiar with any evidence to support that.
So thymol's certainly active against Chrysidia, we know that for sure. These compounds are probably produced by the plant to be broad-spectrum antimicrobials. So, for instance, the Calluna from Calluna is not produced to be bioactive against Chrysidia, it's produced to protect the nectar from yeast or something else, but it just, because it's broad spectrum, needs to be active against lots of species, it's also active against that. One thing that I didn't tell you about today, because I was a bit, because it would have just made it too long, was that some work I did with a PhD student with Mark Brown, we showed that caffeine is biologically active against Nosema bombi. So to the question at the back about should you put a bit of espresso in your hives, I mean, maybe, I mean, I'd be kind of interested if anybody fancied doing some experimental work next year, trying it out, and just seeing if it worked. Why not? You know, what's the worst that can happen? Oh, you kill your yeah, I don't want to do that. No, but seriously, I mean, you know, it's not a bad thing. I mean, there is, you know, good evidence that caffeine you can buy in abundance. So it might be something worth trying, but otherwise, so far, I've not done any work on honeybee diseases. But of course, Nosema bombi and Nosema cirana is they're pretty closely related. So something that would be biologically active against the one would be probably biologically active against the other. But that would be, you know, why not give it a go? Just really kind of a dilute coffee, sugary coffee in the hive. See if you can have a healthier colony, and let me know if it is.
I do a lot of work around planting to promote bee biodiversity. And this goes back to your question of media misdirection and the nonsense that honeybees are going extinct and all of this stuff. It's very hard to fight your way through the thickets when you've got commercial companies who have interests in selling people stuff.
Absolutely.
My big concern is bee houses. People are selling them for a £5 in a garage. Yeah. They're awfully sweet. People are hanging them up, and they are really harmful to bees, a lot of them, if they can't be cleaned, if they can't be well maintained, if people don't have an idea of the fact that they're taking responsibility by doing this, by actually trying to create an artificial habitat. I personally would rather plant habitat that the bees can then manage at their, you know, in their own pace. Has Kew got any guidelines published in terms of the appropriate bee housing, because there's just so much nonsense out there, and the bee bricks that they put in Brighton, which are filthy and full of disease. You know, again, it's this thing of everybody wants to help, but if they're purposely misdirected, they end up doing more harm than good. Yeah.
No, I think you make a really good point. We don't actually. I mean, to be fair, I'm a sort of a bit of an oddity at Kew in that I work on plants and bees. Most people just work on plants or fungi. So we don't have a huge pollinator group. I mean, there's 6 or 7 of us. So we don't have any kind of any guidance at that level. We have a lot of interpretation, and we do share information. We often do outreach days with Reading where we have similar stories. We do have our own bee hotels because they do help us to tell stories. In fact, there was a company called Nurturing Nature, but he's given it up now. Yeah, he just got a bit tired of it already, and people ripping him off, and nobody else seems to have replaced it. He did have someone that was maybe interested in buying up his sort of his kit to make all this stuff, but they were really good. And he was like everyone in the room, I'm sure, he was a naturalist in that he knew what was required. He knew that you needed a certain diameter of hole for different species, for Osmiolaeanas, or but whereas the bee hotels you're talking about is just a bunch of bamboo with holes this big for like a bee that doesn't exist. And it's problematic. But yeah, at the moment we don't have any published guidance. We have had, you know, different. When we got the hive, which was a wonderful opportunity to sort of celebrate pollinators in a more overt way at Kew, I did write a chapter in the book that went with it, and we addressed some of these issues, perhaps not specifically these dodgy bee hotels you get. But yeah, you see them all over, and they're just full of spiders and just falling apart. And I mean, maybe for some, you know, for maybe, yeah, exactly. Maybe they're great for other insects in the end, but they're no good for, you're right, they're no good for bees. Yeah.
I was just saying quickly, I'd love to talk to the lady at the front about bee hotels. That's something that I work with at the moment.
Okay.
But the question was relating to what we were talking about earlier with how specific the chemicals are to different bacteria, and I wondered if any of the compounds actually might have negative effects on the gut flora of some of the bees that you...
Yeah, that's a really good question, and something we haven't yet really investigated as much as we would like. I mean, certainly in the example we gave with Arbutus and the compound unidone, it would seem that the microbiome is involved in activating that compound. But you're right; these are broad spectrum compounds, and they have antibacterial properties, and so you would have thought they may have some effect on the gut fauna. We don't yet know.
In Tanzania, they've recently banned certain honeys because of their nicotine content. This has been linked to a huge expansion in growing of tobacco in Tanzania.
Right.
Is there any biological advantage in the nectar having nicotine in it, do you think?
Well, nicotine also has antibacterial properties and could probably, but I'm a bit surprised because I don't know, do honeybees, I suppose they may rob, because I don't think they can get in, can they? Because they're quite long corollas, do they? Can they get in flowers? But that is a strange problem. And I guess it's not ideal, is it? It's a little bit like the story where you get kind of with it's gone now, Echium vulgari. What do you call it? What's its common name? Echium vulgari? Viper's bogloss, thanks. Viper's bogloss honey, which is obviously one of these that's potentially sort of toxic. It's that kind of same scenario. To the bees, it seems like it's not a problem, but that is I hadn't heard of that before. I'm going to look into that. Okay, thank you.